This is a conversation with Professor Andrea Antal, Group Leader of the Non-Invasive Brain Stimulation Laboratory, Department of Neurology at the University of Göttingen in Germany. Professor Antal is one of the leading electrical brain stimulation researchers in the world, having been a lead author in many seminal ethical and safety papers that guide the field today. In this interview, Professor Antal tells us about some of the exciting developments in transcranial electrical stimulation, using methods such as direct current, alternating current, and random noise stimulation, but also some challenges the field is facing, which is restricting patient access to potentially life-changing treatment. Here is our conversation. Hi, my name's Dr. Trevor Brown, and thanks for joining me. I'm Academy Manager at Neurocare, and thank you, Neurocare, for sponsoring this again to bring it to you for free and allowing me to speak with Professor Andrea Antal today. How are you, Andrea? Thanks for joining me.
Hi, thank you for the invitation. I'm very happy to be here. So it's early morning in Germany.
Yeah.
I'm doing well, yeah, with a coffee here. And yeah, so I have been working at the Department of Neurology at the University Medical Center in Göttingen, and I have been doing brain stimulation for 25 years now.
25 years, and you're actually professor and a doctor, and so group leader at the non-invasive brain stimulation laboratory, Department of Neurology, University of Göttingen in Germany, and so you've had a very I guess interesting journey, because you've been involved in some of the absolute pioneers of electrical stimulation and brain stimulation, like Walter Paulus and Nietzsche. You've been a real inspiration to a lot of people around the world, Andrea. So it's a real pleasure to have you, Andrea. It's brilliant, and yes, you're in Germany at the moment. It's early morning, and I'm in Melbourne, Australia, so it's in the afternoon. So just for people listening, Andrea, I'll just tell them a little bit about what we're going to be talking about today. We're going to get to know you a little bit, Andrea, chat about your education, some previous research, what you're doing in the field of transcranial electrical stimulation. In particular, we're going to focus on your involvement as the Vice President of the European Society for Brain Stimulation. You're also a member at large for the International Federation of Clinical Neurophysiology, which is the IFCN, and you're leading the mentoring program of the European section. Is that right?
Mm-hmm.
Okay,
Mm-hmm.
great. So I'm really looking forward to hearing about those bodies that you work with, and your current work in the field of TES, because we started a lot of work in electrical stimulation has been in transcranial direct current stimulation, but you're starting to branch out into more of the transcranial random noise stimulation and alternating current stimulation. So we're going to hear a little bit about that. So that's just a little bit of a summary for those listening to what we're going to be chatting about today, Andrea. So, but before we get into that, tell me a bit about yourself. Where were you born? Where did you grow up and get educated?
Yeah, so I was born in Hungary in the 60s, so very long time ago, yeah. And I studied biology. So I am not a medical doctor, I'm a biologist. I got my diploma in 1990, and I left the country a few months later because I got a very nice offer from New York, from the Mount Sinai Medical Center for PhD.
Fantastic.
And it was 1990, so and the direction was animal models of Parkinson disease. That time was the MPTP model NOI, mainly in monkeys, and I started my PhD there. I was trained to use electrophysiology, so with implanted electrodes in monkeys, behavioral stuff. And I went back to Hungary in 1994, and I taught physiology at the medical university until 2000. And after I was invited, Germany, Walter Paulus invited me. I was a postdoc, young postdoc, and at first for 3 years, and I stayed here. In 2005, I got my habilitation. This is a German system for teaching. So I learned the language.
Yeah.
And since 2010, I am an extraordinary professor here.
Fantastic! Wow, what an education, and I imagine so. You speak Hungarian; that's your first language, and
Yeah,
then you spent some time in the United States, and so you learnt English, and you learnt English before you went to Germany, and you learnt German, or vice versa.
German, no, no. So I had to learn the language here. So it's. But if you would like to stay in a country, there is no other possibility, just to learn the language. But it's
True,
yeah.
absolutely, but fantastic. Well, what a toolbox of skills you have. Obviously, being educated like that and speaking all these languages. Very lucky to have you. And in fact, when I was doing my PhD and a lot of my previous studies, I saw a lot of your work, and particularly all the work around safety and ethical guidelines. Now, you've worked with some of the pioneers of electrical stimulation, and now you're on the boards of the IFCN and European Brain Stimulation Society. Is that an area of your work which you particularly like, the safety and ethics and regulatory area? Is that something that you do because you enjoy it, or is it something that is just something that the field needs so badly?
Both of them. Both of them. This is the response. Of course, safety is very important when we are using brain stimulation methods, and safety and the efficacy of the stimulation is determined on the surface of the brain. Nevertheless, we apply these currents and magnetic field from outside. And until today, we can just estimate how much current reaches the brain and really what's happening in the brain.
Yeah.
Because very rarely we have, of course, epileptic patients or monkeys with implanted electrodes where we can see what's happening during stimulation. But at healthy subject or patients, when we are treating patients in the clinic, this is really just an estimation. So this is why I think safety is very, very important. And before we started with electrical stimulation, we had already this TMS, RTMS guidelines. It's called Rossi guidelines because they are coming from Simone Rossi. He was the first author and the coordinator writing these guidelines. And we started the first one. It was 2016. It was a big conference in Göttingen, and then we organized a safety workshop for tES. And we just updated this guideline, so it's in progress. Now we had a workshop in Munich in June, and a new low intensity or limited output tES guideline is just submitted.
Excellent. That's really good news for us because we're obviously teaching a lot in Neurocare Academy as well, and so we rely on this work being done by all you guys, and it's so important for the field to help people in teaching roles to teach the clinicians to provide this treatment safely. So thank you very much for working on that because it's a big job.
Yes, it is a big job. I think the guideline has now about 50 co-authors from every other of the world, yeah. So this is the first part of your question. Yes, I enjoyed it and I like it, and I think safety is indeed the most important thing when we are using these stimulation methods. So we should know that what we are doing is indeed safe. The problem is that safety is very, it's very difficult terminology because you know that something is safe from the border when it's not safe.
Yeah, I understand. I
And there is
understand.
a huge variability, of course, because there are different anatomical, physiological factors and other things what we cannot control. And so this was the first part of your question. A second part, yeah, what happened in Europe? The new medical device regulation that is not so new, 4 years old, and the reclassification. Yeah, so we had no other choice, just to work on safety and understand regulation and to protest.
Yeah, yeah. So
So,
you're starting to refer to this 2022 regulation in Europe that reclassified electrical stimulation as a class 3 device, similar to neurosurgical procedures, and so we had to sort of you were instrumental in helping us work through that.
not even that, but before the new MDR, so the MDR, before we have the MDD, this is by the directive, and now we have medical device regulation. And the big difference is that before we were able to adapt the national rules to the EU regulations, and now everything is coming from above.
Right.
And we have a very, very narrow play room and possibility and what to do. And research is some way forgotten in the MDR. And this is one of the main problems. And the interpretation of the law, so 10 ethical committees and 10 different interpretations.
Right. Right.
Even using, for example, neurostimulators.
Mm, mm. Yeah, it's very confusing for
Yeah.
for I guess the layperson. I mean, you're very heavily involved in this, and it's a very in-depth topic. But just if I can summarize,
Yeah.
electrical stimulators were used relatively easily as a non-medical device, but in 2022 they were reclassified by regulatory bodies, and then and that made it difficult for electrical stimulators to be used, I guess, as easily in clinical and medical, clinical and research settings. So you wrote a publication, or you were involved in a publication in 2024 as an opinion paper. It was entitled "The Consequences of the New European Classification of Non-Invasive Brain Stimulation Devices and the Medical Device Regulations Pose an Existential Threat to Research and Treatment." So can you tell us about that paper and what your position was, and has that had any impact on the regulatory proceedings?
Yeah. So as I mentioned, the first step was the NDR, and the different kind of interpretations from the ethical committees. And the main problem is here that the ethical committees are looking for this terminology, that intended purpose. And this should be in the book, what we get from the company. But this is different from indication, because the indication is coming from the medical doctor. And unfortunately, yes, in lots of companies certify the stimulators using or treating just one disorder. For example, fibromyalgia, depression, or chronic, or something like this. So that is a definition. It's not neuromodulation. That is, for us would be great that something like neuromodulation or transcranial stimulation or something like this. But this is not the case. And the ethical committee is okay. It is just for fibromyalgia treatment. So if I would like to treat patients using exactly the same protocols in a clinical research setting, because we would like to know that how does it work in low back pain patients, they say that no, it's not possible. We have to submit the proposal to the National Regulatory Agency.
Right.
And it's time, of course, more money, because under the MDR, we have to pay insurance for even for healthy subjects and patients and everything. And so it's a very similar system like the approval of medication, pharmaca. So they try to copy this pharmacological approval system, what is working very well in Europe. But medical products are not really pharmacas. So this is the first problem, that much longer approval process. It's expensive and extremely bureaucratical. So I have submitted already one proposal treating cancer pain patients, and I had to submit 150 attachments. Just getting an ethical approval here in Göttingen, and it took us for a year.
Yeah.
So it means that it's very difficult to accommodate this time, for example, in a PhD project, or even in a simple grant, because it's a long time, lots of money. The grant agencies are not willing to pay the extra cost, not yet. And we have significant delay in research, in treatment, because patients are waiting for the new protocols, the new treatment options.
So that's that. If I understand correctly, these regulatory changes are really restricting your ability to do your work, right? To do your research and then make these clinical applications possible and accessible to people.
Yes, exactly. And it started already 21 May. And after, the top of it was, of course, the reclassification in 22 December. It was really unexpected. But this is just for the non-intended use. So the non-medical purpose. The problem is that research, what is research? How can you, it's a medical or non-medical?
Yeah.
Because we are between, or neuroenhancement. Of course, in young, healthy subject, it's neuroenhancement. But if you would like to treat elderly, not even with MCI or early Alzheimer's disease, but elderly who are starting to forget things, is it treatment or neuroenhancement?
Yeah.
So that is really the border. And this was really just at the top of everything, because the reclassification is based on safety issues and statements. And if you read the law and the statement, they are absolutely incorrect. So this is the main problem. And if the ethical committee reads this statement that abnormal brain development, anxiety, whatever it is, so at least 10 different symptoms and adverse events, even adverse events what we have never experienced.
Right. Yeah. Yeah.
And there were 5 papers, so they listed 5 papers because 6 EU member states submitted this letter that reclassify brain stimulation devices to class 3. 5 papers, and these papers were published at least 10 years before 2022. So they ignored the research what we have done during the last decade and the facts. For example, I have never seen an epileptic seizure using TDCS or TES or TRNS during the last 25 years. And with TMS, I have seen one.
Yep.
And we have simulated several thousand subjects here. So, and this is why we started to protest. So we need regulations. I am not against regulations. We need regulations. We need statements. We need law, but they should be. So it cannot be that the interpretation is the law. It depends on the ethical committee or national regulatory body. The law should be law.
Yeah, and it should be based on recent research, not 10-year-old research, of course. So, yeah, fantastic. And so, how are things moving now? Are things moving forward? Are you starting to get some better accessibility?
Yes. So we started really a protest action in whole Europe. together with the European Society for Brain Stimulation, where I'm vice president. And indeed, so European, Middle East, and Africa chapter of the IFCN. This is the biggest chapter of the IFCN with joint efforts, and we submitted protest letters and everything but what you can imagine, collecting signatures. And what happened? So the ombudsman decided that, they have to rediscuss this issue. And indeed, the medical device coordination group now is collecting data, as with a share group who is collecting information and data about safety of the brain stimulation devices. They will do a risk management and description of these devices. And if we are lucky, they can amend the law. But unfortunately, until it's not amended, we have to live with the with the recent regulations. So there is no other possibility.
Right, you're still in this situation where you're attaching 150 documents and spending a year trying to get an approval for a research project, I assume,
yes,
and that, and as you said before, costs a lot of time and costs a lot of money.
Yes, and what they really did not consider that there is a huge difference between the approval of pharmaca and then approval of, for example, a medical product when we use the medical product for treatment. Because pharmacological companies can support and sponsor the whole development from preclinical until phase 3 studies. Nevertheless, concerning medical products and mainly brain stimulation devices that are expensive, for example, a TMS device, one device is 80,000. It's almost impossible to sponsor the whole process when we are developing new protocol, new devices. And therefore, we have smaller studies. So 20 subjects stimulated here, 20 subjects there, and these big phase 3 studies are still missing. And therefore, we have less data on safety, mainly using the newer, newest methods and protocols. And one more thing, in research, we have to adopt the protocols continuously. This is why it's called research,
Of course, yeah.
And
it has to be independently replicated and shown in multiple settings. Yeah. Well, I think in the field of neuromodulation, I think it's great that we have so much communication like this, and we see the limitations of neuromodulation research from a financial perspective, but also from the perspective that it takes patients quite a long time to go through treatments. They might do 30 or 40 sessions, and so this limits the sample size that we can obtain also in neuromodulatory research, and it also is very costly and time consuming. So we need to sort of collaborate between us, between the different centers, to increase the sample size and create these studies.
Yes, I agree. So this can be solved only with joint efforts. Yep. And this is, so the new guideline is a joint effort of the IFCN and ESBS. So I brought together two societies here because the IFCN has a special interest group for brain stimulation too.
Excellent. Excellent. Well, thank you. Well, I must thank you on behalf of the field of neuromodulation. I mean, we have clinics, we have a lot of clinicians who are using neuromodulation and treating a lot of very happy patients who have access to this. And it wouldn't be possible if we didn't have people like yourself who are fighting these battles and doing all this hard research. So thank you for that. And so since we're on the topic of ethics and regulatory issues, you also published or have been involved in an ethical debate surrounding do-it-yourself electrical stimulation products that have really come to the fore over the last 5-6 years in particular. They're being used for performance enhancement, just bought off the internet, home-based kits, not necessarily have been approved from a regulatory body or have been shown to be safe. What's your involvement there, Andrea, or can you expand on that for me?
Yes, so It was really a surprise for me that time. So there was this movement was started already in 2008, and even in Germany, that private persons started to build electrical stimulators at home. I think they started with magnetic stimulators too, but it's more complicated, and it was not possible. And indeed, these do-it-yourself stimulators are used for gambling, so improve working memory performance. And what is sometimes dangerous, I got phone calls from parents that they would like to treat their children because the child is not good in mathematics or whatever in the school. And what is my opinion and where to put these two electrodes? It's and how to do that. And so I'm really against using do-it-yourself stimulators. So don't buy the internet, Amazon, eBay, or whatever. And I have to tell you, I am against a little bit. Or we have this direct-to-consumer direction from companies to here I'm a little bit ambivalent, I have to tell you, because I can understand the patients, that patients are looking for treatment options, and they really would like to try everything, and I support this direction, because indeed this is why we are doing our research and clinical research work. But on the other side, that everyone can buy a stimulator, something like saying that, okay, I have depression and I would like to get this stimulator, and you get it with a nice video guide, and just to, so this is some way. So I think this case is medical doctor or a prescription or something should be there, it should be involved.
It needs to be at least followed by a clinician of some
Yes.
description to have a look
Yes.
at outcomes and officially assess the outcomes of things, and obviously maintain safety and do no harm from the outset, of course. Yeah.
Concerning the safety side, I think using electrical stimulation, the worst is that the skin is burned, something like this. It happened many times.
It does.
Yeah.
It's a relatively safe treatment. If as you said before, seizures are very, very rare in TMS and practically unheard of in TES and electrical stimulation. Like you, we've treated thousands of patients and we've never had any seizures. But if things are not applied properly, you can burn the skin. There are some issues there if you don't use saline solutions. So people that buy things off the internet assume that they can't harm you. I think there's an assumption there that if you can get something easily, well, then it's not as dangerous. And so it is very, very important to be overseen professionally by a clinician who knows what they're doing, has been trained adequately to use these techniques. So look, I think we are also advocates of not using do-it-yourself tools, right? Of course, in neurocare, and you should definitely have some oversight from a clinician if you're going to use electrical stimulation. But look, thanks for that, Andrea. Have you got anything else to add on the do-it-yourself side, the ethical side?
Yes, I would like to comment one point, that I really support the remotely supported or remotely supervised home stimulation. So this is the new direction, because indeed we save time, we save money, and it's more comfortable for the patient.
Yep,
So
exactly.
I
I
really,
mean,
really like it. So it's and we have these stimulators, and I really, and indeed I think it's better maybe. So I can understand the patient side too. Someone has this depression, and sometimes patients do not want to go to the doctor, or because it's still a stigma, a mental disorder, whatever. So I can understand this side, and maybe, but some way a medical doctor in that case should be involved in the whole process. And I think
Yep.
home stimulation is really a great option for patients who don't want to come each day to the clinic, but it should be some way supervised.
Yeah, look, it's a great point that home-based treatment, I guess, is the way of the future. And when you have remote areas, people who cannot get to a medical doctor,
Yeah.
it's great to have the accessibility to have a home-based treatment, but it should be supervised in some way, of course. Um, Look, thank you very much, Andrea. I think home-based treatments are moving forward, but one of the safety issues is that there's a lot of variability in response to transcranial electrical stimulation, and that's not because of the variability of the devices. The devices are very safe in that they transmit a very consistent direct current of a particular ampage or frequency or whatever's being used. But there's a lot of inter-individual variabilities like genetics, like your current brain state, like all sorts of hormonal issues. So when you're being treated with neuromodulation, it's not easy to predict your absolutely personalized experience to it and your outcomes to it. And so that's why it's really important for it to be overseen by a clinician.
Yes, I agree. Concerning, so when we are using TMS or RTMS treatment, there we always have something like a motor threshold measurement and we adapt the intensity. But when we are using TES, TDCS, TACS, we use usually this 1 milliamp, 2 milliamp, 4 milliamp intensities, so for everyone.
Yep.
And then we never know how much current reaches the brain or deeper brain areas of what's really happening. And indeed, variability is very important. Partly is coming from the anatomy, the thickness of the skin, the bones, the cerebrospinal fluid, what conducts very well. And other part, what you mentioned, the state of the brain. However, so this variability is there. We have to live with this variability. We have no other possibility. And there are factors what we did not consider before. For example, the excitability of the brain is during the whole day is changing. So we have different excitability in the morning, noon, late afternoon, depending on, of course, what we are doing, but depending on what kind of type, what kind of chronotype we have.
Exactly.
And it's very rarely considered in the studies. Or, for example, hormones. I teach
Yep.
neuroendocrinology. And if you consider that, for example, the female sex hormones, so estrogen, progesterone, they have a huge effect on cortical excitability or cortisol, stress hormone.
Yeah, look, there's so many different variable factors there that it's too hard to account for all of them and predict the response. So we're still working on trying to understand all of these, aren't we, Andrea, so that we can actually personalize treatment. I mean, with TMS, we're able to personalize the dosage a little bit using this motor threshold. With TES, we're not able to do that, and so we generally just give a one-size-fits-all type of protocol to everybody. But we're starting to find some biomarkers. For example, I've seen some research on theta peak, which is used for TES for memory consolidation and to be able to actually dose the electrical stimulation according to the theta peak. So is there any recent research that you know about or that you're working on that may help us personalize the treatment a little bit?
Yes. So nevertheless, so concerning personalization, it has really two sides. So one side, we would like to understand how does the brain react when we are using neuromodulation. Nevertheless, on the other side, there are the medical doctors, the treatment side, and they want to keep it so simple than possible.
Yeah.
So they have no possibilities to put the patients in the MRI, fMRI,
Yeah.
or even sometimes to measure even the EEG.
Yeah.
And indeed, there are electrophysiological biomarkers that when the theta activity or alpha activity is higher there and that the patients would react better. Or just very simple, so more current reaches the gray matter, better effect. Sometimes there is a very simple relationship, but in small number of subjects, so it's not like 300 or 400 subjects, but just 20. Sometimes just healthy subjects, and healthy subject, the measurements in healthy subjects can be very different from the measurements what we can consider in patients.
Yeah.
Or
Yeah.
younger and older. So what we are doing is puzzle. We putting this part here, that part, but we do not have this nice whole picture. And concerning personalization, yeah, it's very, very important. And we have running studies with the personalized intensities with the current flow stimulation before we start to stimulate.
Yeah.
But it's financially really, so it's much more than just putting on 2 electrodes and using this 2 milliamp intensity. And I still cannot tell you that it's worth to do and invest the time and energy in the personalized direction, or it is really good and acceptable then we use 2 electrodes with 35 square centimeters and we stimulate the half of the brain. So I don't know.
Yeah, I mean it's a hard question, isn't it, to say whether we invest the cost in making something a tiny bit more precise without much clinical benefit to resulting from that investment. But you mentioned just before that we have different distances from the scalp to the cortex, and this particularly in the elderly, there's frontal atrophy, and so there's this sort of variability that we need to take into account. But in fact, you're actually working on with your team some research with the elderly at the moment, working memory in the elderly, visuomotor learning in older adults. Can you tell me a little bit about the exciting research you're doing at the moment with the elderly?
Yes, so in Göttingen we have now 3 different running studies using alternating current stimulation and RTMS to treat healthy elderly, and we try to go in the direction mild cognitive impairment. But these elderly are not diagnosed. But based on the test, they can be diagnosed with MCI, yeah, something like this. And one study we just finished is measurement of dual task performance using stimulation, under stimulation, so tES and tES, 10-day stimulation, and in this study we had 60, 70 elderly subjects, and the data analysis in progress. The second study is running. Here there is a huge development, but because we are not using anymore like a computer task, but we are using virtual reality.
Wow.
And here the task is a little bit personalized too, because it's an NPEC task, but they can choose that something like they are in the garden and they are watering flowers, or they are in the supermarket. Wow. And they are doing shopping, or they are at home and they are looking for keys or different things that are hidden in the apartment. because it's not so boring.
Yeah.
But you know, this card what we are using, or letters for NBAC, and we are using like adaptive NBAC, so we can
Right.
make the task more difficult.
Right.
if they,
And just for those listening, an NBAC task is a working memory task.
yes,
Right?
working memory task.
Mm.
And here we use tES,
Yep.
so alternating current stimulation, more parietal stimulation. And concerning the rTMS study, this is really a new direction, because here we consider the heart rate variability change as a biomarker.
I see. I saw quite a bit of research recently on heart rate variability and to do with TMS from Martin Arndt's team, and using the heart rate deceleration and showing that in fact some of the sham coils in TMS were not particularly inactive, that they were actually modulating heart rate a little bit as well.
Yeah, so it's very, in the TMS, rTMS is very difficult to perform a sham stimulation.
Yeah.
And we have a dry G plastic coil,
Right.
using that, and we recorded exactly the sound of the real rTMS machine. But still not the same. Nevertheless, we have parallel groups, and we can say to the subject that, okay, it's not like placebo and real stimulation, but you can get other type of stimulation. You know, if the ethical committee allows you to tell it the patients, you can do that. But indeed, with the rTMS, is more difficult. You can use electrical stimulation under the coil, but it's still a different feeling than the real stimulation.
So that makes your research very difficult, Andrea, doesn't it? Because it's not only a placebo effect. You have a sham, which is actually trying to produce a sensation on the cortex of electrical stimulation. So you may even be stimulating, for example, the trigeminal nerve, or you may be stimulating other nerves on the cortex that are actually producing a real effect, which makes the difference between the experimental group and the control group. It makes the difference even smaller, because the control group is actually receiving some sort of active component in the sham stimulation. Maybe if we have open label studies that are showing the real effect of neuromodulation, they'll be quite a lot larger than the randomized controlled trials that you're doing.
Yeah, it is true. And if you compare the different sham conditions that are pre-programmed in different companies, they are really different. And so in one of the companies, the sham condition, during the sham condition, subject patient get 3 times more or higher dose than using another stimulator. So it's therefore I suggest not to use only sham, but use an active control condition. And mainly it's very important when we are using, for example, tES, like another frequency, another stimulation montage. Because during tES, it's not just the skin sensation, but the phosphine perception is there. If you use if you use an EEG stimulation frequency window, and so. And indeed, it's good that not to switch on the stimulator, maybe just putting on the electrodes and have a condition like this, or to compare it to a non-stimulator. So there are so many possibilities. You know, it's a never-ending story.
It is, and we're getting into some of the very in-depth aspects of research now. But it's important for everybody who's applying neuromodulation to know that there's a lot of very, very detailed research that goes on before these things reach the public in the clinical population. So it's nice for us to understand all these things as well, but to know that we have some very smart people that are looking after these things for us, and before they get applied in a clinical setting. So you're actually using. You've used tDCS, direct current stimulation, for a long time. You're involved in tMS, transcranial magnetic stimulation. The random noise stimulation and the alternating current stimulation. Now, this has started to generate a little bit of interest recently. Why is that? What potential is there for RNS and ACS above the tDCS?
Yeah, so we started the tES, tRNS research in 2005, 2006. And that time our main objective was that, okay, the brain is working with oscillations. And before we use this direct current stimulation, very, very oversimplified, anodal, cathodal, and this direction, that direction, facilitation, and inhibition, but it's not really true, but this is the oversimplification. And we believe that, okay, so if patients have abnormal oscillations, and there are plenty of disorders with abnormal oscillation, if you think about like schizophrenia or Parkinson's disease, we just have to apply the correct frequencies, what would work from outside with a high intensity, and we can reset the brain and stop this maladaptive oscillation, and it did not work. But this was the main objective where we started. And after indeed we discovered that, okay, if the frequency of the stimulation what we would like to apply is very far away from the frequency what is in the brain, what is there and exists, we have to use higher intensities. And these frequencies usually not just there and they are alone, they are changing during the task according the state of the subject, and they are coupled like theta gamma coupling. So so there are plenty of things what we have to look for and check before start. But indeed, this is a new direction, and random noise stimulation. Yes, so this is like a type of tACS. So plenty of frequencies, a bunch of frequencies together. Until today, I don't know how does it work, but I have to tell you, it's working. So there is a huge effect of tRNS. Advantage is that the skin sensations are not so strong than during tDCS. There are no phosphine perception like during tACS, and it has a very strong facilitatory or inhibitory effect, at least when we use in rest, and we measure the amplitude of motor evoked potentials. Compared to, for example, another tDCS, motor cortex stimulation in rest, if you use the same parameters, the duration of the after effect is longer and somewhat stronger. Right. But concerning tRNS, we do not have really animal studies.
Right. Okay. So what I'm understanding, Andrea, is that for many years we've tried to do tDCS by increasing excitation or decreasing excitation in a very linear fashion, and with tACS you've described that we tried to take a frequency that was abnormal, for example, I don't know, 10 hertz alpha, and tried to make 9 hertz alpha and tried to push the frequency in a certain direction, but these things haven't worked. Major successes. Potentially we go so far away from the brain's oscillating frequency that we disrupt the circuitry in some sort of way that allows it to reset itself. I guess is that the way you're looking at things now, to just sort of reset things?
Not really. So I think I learned a lot too during the last 20, 25 years. And I really realized that how much we, until today, do not understand.
Mm,
So I'm going with the whole group in the direction is more like a holistic treatment.
mm.
Or even the holistic neuroenhancement direction. So, do you think that if we stimulate just one part of the brain, the other part does not start to compensate or do something? So, if we do something in the brain, something happens in the body, and if something happens in the body, we have an effect on the so. Um, and in the brain, we have inhibitory and excitatory neurons.
Yeah.
And what we really change the inhibition or so excitation inhibition balance, but it's continuously changing in the brain, and we can modify this activity from outside.
Mm, mm. Yeah, look, I completely
But
understand what you're coming about, because if we try to guide the brain, it's sort of an arrogant perception that we can lead the brain and that we know what's going on, whereas there are compensatory mechanisms, there are other
yeah,
systems that will take over. So if we try to, I guess, reset a certain section and then allow the body to do its own thing, because the body is the best. The best carer of itself, I
yeah,
guess, from a holistic perspective, like you say.
yeah. So and we have not only, for example, transcranial based stimulation, but now we have spinal cord stimulation, and so there are so many possibilities, and you can combine spinal cord stimulation with transcranial brain stimulation. So, for example, if you think about ELS treatment, um, that indeed the problem is that until we develop a protocol, and usually we test our protocols on healthy subjects, and we start with patients, there are so many steps and so much time. And sometimes we are going to the wrong direction, wrong electrode position, parameters. So what we really need, like biomarkers, um, and with the help of like so diagnostic biomarkers, I would say diagnostic biomarkers, that we can say that okay, we can predetermine if the stimulation will have an effect when we stimulate this subject or patient or not, and we don't have to wait until 10 stimulation session to see that okay, nothing happened.
Yeah, yeah, yeah. Look, Andrea, you've been very generous with your time. I'm just mindful of not taking up too much of your time. I have one extra question, if you don't mind. Because of your expertise with the elderly, and there's been a little bit of interest in using electrical stimulation for long COVID and brain fog. How do you think the field's progressing there, and is there much potential there for use of brain stimulation for long COVID and I guess with mild cognitive impairment and brain fog?
Yes, there are running studies, there are published studies, and I'm convinced that, for example, the treatment of fatigue, when we are using the protocols what we use for MS fatigue treatment, they can successfully applied. I have to tell you, so we submitted a proposal already 2-3 years ago. So at the beginning of the COVID phase, it was rejected for the treatment of brain fog, cognitive impairment due to COVID and fatigue. And after I did not follow it that way because, and again, the problem is here, it will be an MDR proposal because we do not have stimulator with intended medical purpose like fatigue or COVID or brain fog.
I see.
And
I see.
so
So
regulatory,
it's
so
holding
yeah,
it's holding you up. That MDR and that regulatory issue
yeah,
is holding you up for using devices for that purpose, because the
yeah,
intended purpose may be depression or something else. I see. No, it's a problem. I guess it's hamstrung you a little bit in the past, hasn't it? So if we were to think about your 25 years as leader of the Brain Stimulation Lab in University of Groningen, where do you see the field heading over the next 5 or 10 years? Where is the greatest potential for electrical stimulation, do you think? Last question.
We have to go deeper. So developing protocols that we can reach subcortical areas. One possibility would be this temporal interference stimulation, or like combining different type of stimulations or electrode position and switching on and off the stimulator, or so I hope that the field is going to the home stimulation direction. That is more reachable for patients and general practitioners. Some way we have to do something with the reimbursement. So the insurance companies should pay for the treatment because, for example, in Germany, this is now the treatment of the rich people. Unfortunately, and we should harmonize this reimbursement system, at least in the EU. This would be my plan. For that, we need a training and certification system, what would be very, very important. And we started this direction already.
Great.
Yeah.
All very good advice, Andrea. So, look, thank you very much for your time. You've been very generous. It's fantastic to speak to someone with all your knowledge and experience. So it's been a pleasure to speak with you, and we'll try to keep in touch and spread the word and help collaboration and help training and any way we can here at NeuroCare. And we'll stay in touch with our training to try to help the certification process for you. So it's been a pleasure speaking to you, Andrea.
Thank you.
Thanks for listening. We hope you got something out of this conversation, and maybe it sparked an interest in developing your skills as a therapist. If you're interested in taking the next step, head to lms.neurocaregroup.com to access a range of professional online training courses in neuromodulation techniques such as TMS, TDCS, and neurofeedback. If you're looking for where to access these therapies, head to neurocaregroup.com to find our network of clinics throughout the world. We'll put these links in the description below. You might also be interested in listening to our other conversations I've had in the series. Be sure to follow our channels on YouTube, Spotify, or however you subscribe to your podcasts and listen to other conversations I've had with a range of experts in the field.